Facilitator. So I'm joining remotely from the lands of the Songhees people in the settler community of James Bay, built atop Hoosakum, in the traditional territory of the Swangwan family group. I really appreciate this opportunity to share my research with you. I hope not to bore you too much. I'll be using specialized terminology, and seeing as there are a few of us, you can interrupt me at any time. Some of the terminology I'll be using are things like ecohydrology, phenology, spectral phenologies, and properly resolved remote sensing. Most of the terms I'll introduce and explain, but if you need me to translate anything, please raise a hand or just interrupt if you need clarification. And even if you want me just to stop, say stop. I'm fine with that too. Hopefully I'm able to communicate the importance of the work that we've done, but that is not guaranteed. All right, so despite their importance, especially to climate change, there's a significant uncertainty around peatlands because their extent, status, and dynamics have never been globally mapped with sufficient accuracy. There are also issues concerning the reliability and accuracy of peatland remote sensing products. In this image, we're looking at a remote sensing product showing phenology metrics for Burns Bog. In this case, an average spectral vegetation index at a 500-meter spatial resolution. I'll be explaining most of those terms later, both phenology, what spectral vegetation index means, and what a spatial resolution is. I mainly want to draw your attention to the size of those grid cells as they overlay Burns Bog, which we're looking at underneath. In this image, you can see the Vancouver landfill near the bottom of the image, and then my actual field sites just above the letter U in the word uncoupling. Because of the issues concerning the accuracy and reliability of peatland remote sensing products, I want to highlight a couple themes in my presentation. The first of these themes is what I term spatiospectral uncoupling. This uncoupling is a function of spatiotemporal scale and the interaction with spectral resolution. So you're talking about the resolution of both your time, your space, and then the spectrum of light that we're looking at. This image here is a half square meter sphagnum plot taken during summer drought. You can notice the diversity of the false colors and the associated spectral vegetation indices in the legend. It's important to think of a comparison with the average index value from the 500 meter squared resolution in the grids that we just saw in that remote sensing product. Both of these images are from the same time period and come from the same area of the bog. The only difference being that the spatial scale achieved between the two images. It's important to highlight the large differences in scale between satellite-based remote sensing and the multispectral imaging that we have carried out. I would also like to highlight a theme that is referred to as phenospectral bias. I noticed this bias in research that relies on remotely sensed products that favor vascular phenologies, meaning the life cycles of vascular plants. The emergent vegetation in this photo is bog cranberry. You can see the green with the red tips on it. If you want a pure and unmixed signal of reflectance from the moss layer, this would not be an ideal time to capture an image. So, but why are phenospectral bias and spatiospectral uncoupling even worthy of our attention? You may be asking why either of these themes are important. Why should we care about spatiospectral uncoupling when moving between scales? And why is it important to look at the phenospectral bias when employing near and remotely sensed products? Maps give us some context. This map shows the global distribution of peatlands, though I should say that not all peatlands are sphagnum dominated. When I refer to peatlands, I'm referring to natural areas covered by peat, with peat being partially decayed plant material that accumulates under waterlogged conditions over long periods of time. Some terms commonly used when referring to peatland types are peat swamp forests, those in Indonesia, where the orangutans call home, as well as fens, mires, or bogs. Peat is found around the world in permafrost, in coastal mangroves, beneath tropical rainforests, and then in arboreal forests here in Canada. However, looking at the legend of this map, it seems that these are the regions where peat can occur, but what do we know of their actual extent? Looking at this map, it's important to remember that it wasn't until 2016 at its World Conservation Congress in Hawaii that the International Union for the Conservation of Nature agreed to a resolution that recognized the importance of peatlands and their crucial role in climate change mitigation. Though these ecosystems cover only 3% of the Earth's surface, peatlands contain more carbon than all of the world's vegetation combined. That's including forest. It was also in 2016 that the Global Peatlands Initiative was formed with the stated goal of saving peatlands and mapping their extent. Why are these ecosystems, which include bogs, mires, fens, and swamps, so important to global carbon balance? Primarily because they are the most space-effective carbon stores of all terrestrial ecosystems. A 15 centimeter thick peat layer contains per hectare more carbon than a high carbon stock tropical forest. A conservative estimate puts their storage at 550 gigatons of carbon. That's twice the total of forest biomass, which cover 30% of the Earth's terrestrial surface. So a more recent estimate of just northern peatlands, which would include most of Canada, reckons that they alone store the equivalent of 1,055 gigatons of carbon. So the models are getting better. That's over 1 trillion tons. To put that in perspective, the total atmospheric carbon pool is estimated at around 750 gigatons. As peatland evangelist and a mentor of mine, Hans Houston, has explained it, in a forest, looking at this image above, there's space between the trees. On the right, in a peatland, it's just wall-to-wall carbon. These visuals should help us understand the carbonaceous reality of peat soils. You trying to do either of these things in mineral soil with low organics, it would just be impossible. You can't fall into it and you can't stick your arm into it. Yet globally, these vast carbon stores are under increasing threat. This is a palm plantation in Indonesia, the home of the peatland forest where orangutans are resident. In 2017, a PhD candidate named Greta Dargi at the University of Leeds published a paper that estimated the tropical peatlands of the Congo Basin to be the largest in the world. Many refer to the peatlands in the Congo Basin as a ticking carbon bomb. Political leaders of the Democratic Republic of Congo and the Republic of Congo want to see the benefits that development will bring. But development will damage the integrity of these massive carbon stores. We know this because it's already happened in Europe, in Indonesia, and in the Great Central Valley of California. Cobalt, one of the so-called gems that is so important to high-tech energy transition, is extremely plentiful in the Congo Basin. Gems refer to green economy minerals such as cobalt, aluminum, lithium, rare earth elements, and nickel. The carbon-centric tension over the development in the Congo Basin has a peatland twin much closer to home. This map shows the potential distribution of northern peatlands based on what's termed the Holdridge Life Zone system. But what is the extent of these crucial waterlogged ecosystems in Canada? Pay attention to the green parts of this map. These are bogs. This map is showing the total organic carbon mass for each polygon in the Canadian peatlands database. Why am I sharing this? Look at the areas with the highest total organic carbon mass, the greens and blues on the map. What are those areas? They tend to be bog ecosystems. This map shows the total organic carbon content for each of those polygons from the same database. Thinking back to the bog distribution map of Canada, the most carbon-dense ecosystems are bog ecosystems, as measured by kilograms of carbon per square meter. For example, 1 square meter of peatland from the Hudson's Bay lowlands, the second largest peatland in the world, holds approximately 5 times the amount of carbon as 1 square meter of tropical rainforest in the Amazon. But there's a strong bias against bogs, and it still runs deep. In Scotland, a place known for its bogs, a politician looking across the flow country, where somewhat similar research to mine has been carried out, said, well, it's Mamba country, isn't it? Mamba meaning miles and miles of bugger all. There's certainly miles and miles of something in these ecosystems. New estimates for Canada's terrestrial carbon store have come in at 384 billion tons of carbon. It's miles and miles of nothing, not really. Now we're looking at a map of Canada's version of the Congo Basin, those Hudson Bay lowlands in Northern Ontario. These areas are vulnerable because they coincide with areas that are of interest for resource development. Our federal government's stated goal of achieving net zero emissions by 2050 would depend on global carbon sinks, including peatlands, absorbing about half the carbon dioxide released by human activity. The consequences of losing more of these carbon sinks and potentially turning them into yet another source of atmospheric carbon is something we haven't totally taken on board. Peatland carbon sinks become carbon sources through forestry, drainage, road building, seismic lines, and mining, basically any activity that impacts the integrity of these wetlands. It's mining that has many Treaty 9 and James Bay Treaty First Nations concerned about the future of the area they know as the breathing lands. Premier Doug Ford recently announced they would like to see the development of Northern Ontario's Ring of Fire region. He wants to see this area mined for those gems, those green economy minerals. It is telling that peatlands are not included in Canada's national inventory of emissions and contributions to reducing climate change under the Paris Agreement. In January of last year, Mushkegowuk chiefs called for a moratorium of Ring of Fire development. In April of last year, Attawapiskat, Forest Albany, and Neskantaga First Nations all declared a moratorium on development in the Ring of Fire. And this past January, the chiefs called for a definitive answer on development from the feds. They want input into the terms of reference for the regional impact assessment of the area affected by Premier Ford's critical mineral strategy. Speaking to the motives of the provincial government and the mining companies, Chief Robert Nakagi of the Fort Albany First Nation said, at the end of the day, they just want to attack any mineral that's sitting in the backyard of the First Nations. I think it's notable that the breathing lands are only called the Ring of Fire because a powerful mining executive was a fan of Johnny Cash. So when we hear about threats to peatlands and bogs, we often think of fire. This is an image of them water bombing a recent fire at Burns Bog. We hear stories about the freakish zombie fires that spread underground, popping up here and there and vexing the wildfire crews. But one of the biggest threats to peatland carbon storage is actually drainage. Drainage has drastic effects on the carbon cycle of northern peatlands. When peatlands are drained, they become unstable. When the eco-hydrological balance of an ecosystem conditions is compromised, these systems quickly switch from net carbon sink to carbon emitter. Although only 15% of the world's peatlands are drained, which is less than 0.5% of the Earth's surface, they are estimated to release at least 2 gigatons of carbon a year, which works out to 5% of all global anthropogenic emissions. In case you're not familiar with common measures of sectoral or territorial emissions, that is what's commonly referred to as a lot of emissions. It can help to think of peat as if it was pickles. When you remove the conserving water, the organic matter rots away and decomposes. For every 10 centimeter lowering of the water table below a peat surface, it has been estimated that on average, 5 more tons of CO2 equivalent per hectare are emitted. So according to many, the drainage of peat and the associated microbial oxidation of peat are more important than the catastrophic fires in terms of the overall peatland carbon balance around the world. Under waterlogged anaerobic conditions, decaying peat decomposes slowly. Sphagnum mosses account for roughly 50% of the peat stored in northern peatlands. And as a genus, sphagnum is estimated to store more carbon as biomass than any other genus of plant. With drainage comes exposure to air, and these aerobic conditions accelerate the decay of this peat. The lost organics are mainly converted to water and carbon dioxide. In a sense, as peat decomposes, the land is vaporized and subsides, just like the land that disappeared from under this World War II bunker. So you can see there, the land used to be right at the base of it, and it's, probably 12 to 14 feet, it's receded. In the far north, peatlands caught under the melting permafrost are at the mercy of general climate policies and global emissions. In more temperate climates, such as ours, in Vancouver Island and the Lower Mainland, the restoration and rewetting of peatlands is considered a comparatively cost-effective carbon reduction method. Researchers seek a better understanding of the contribution of temperate raised bogs to global heating, whether carbon is lost or not assimilated and sequestered. It's important to remember that these landscapes are dominated by non-vascular plants. They are sphagnum-dominated ecosystems, and sphagna are the central component of the peat accumulation process. The slow and steady accumulation of large amounts of carbon in peatlands is a function of the recalcitrance of sphagnum species. This decay resistance is enhanced by the acidifying and waterlogging tendencies of sphagnum, which actually engineer these anoxic and acidic conditions which prevent further decomposition. The carbon balance of peatlands is a function of below ground carbon storage, which is strongly influenced by the sphagnum productivity. As these non-vascular plants lack stomata, sphagnum mosses access the water needed to assimilate the carbon via capillary rise, which is dependent on depth to water. Peatlands are shaped and driven by the unique ecology of these tiny non-vascular plants. Sphagnum productivity is central to peatland carbon dynamics, and sphagnum photosynthesis is a better proxy for sphagnum productivity than traditional growth metrics like growth in length or growth in height. In my research study, I was interested in achieving a better understanding of the when and the how of sphagnum productivity, and how both are related to depth to water along what is termed the hummock to hollow gradient. I wanted to better characterize and describe this relationship at our field site in Burns Bog. The study site is located in the southwest corner of Burns Bog, which is a roughly 3,000 hectare maritime raised bog. Burns Bog sits near the southern limit of the climate envelope for bogs of this type in North America. So, as climate changes, we'll find that these bogs might not be able to exist in the locations they were. Here's an example of what development and climate have done. This slide gives a sense of the historical extent of bogs within the Fraser lowlands. And this slide gives us a sense of what bog ecosystems remain in the Lower Mainland. So not as many. Burns Bog, still portions of it. My study site is comprised of 6 fields that were prepared for cranberry farming in 1998. At the time, the entire living layer of peat was cut over. The area that was cleared covers approximately 30 hectares, including the ditches and the bulks. Bulks being where they've moved the material from the ditches and created almost like a berm. This multispectral imaging study focused on 2 of the southern fields. You can see them there. The fields are roughly 400 meters long and cover around 10 hectares in total. When I say that I was interested in a better understanding of the when and how of moisture-mediated sphagnum growth, that's in reference to the idea of growth versus productivity. These are inherently ecological questions. The assimilative capacity, i.e. the sequestering potential of sphagnum-dominated peatlands, is dependent on the ecological integrity of the system. The depth to water table is a central determinant of peat-initiating eco-hydrological processes. Interesting aside, coyotes in the field like to chew anything that human hands had touched, which meant that the piezometers and caps were a favorite target for their chewing, which meant that I had to build coyote-proof collars for some of our instruments, which did get chewed on, but this kept their little maws off the important stuff, which preserved the data. It's important to remember that carbon assimilation and ecological integrity are not independent of each other. You store carbon if you have a healthy bog, but more importantly, you prevent carbon loss by way of an integrated peat-forming community, and that includes both the plants and the microbes. Here you can see a simplified diagram of our local eco-hydrological gradient. The typical microsites along the gradient are labeled. We've got mud bottoms, sphagnum lawns and carpets, sphagnum hummocks, cushions and hollows, as well as the resident biocrusts we find in some hollows. Along with the microsites, the high and low seasonal water levels are hashed out, as well as 2 broad plant functional types, the graminoids and the ericoid shrubs. But most importantly, you can see the relative position of the 3 indicator species of sphagnum moss that we monitored, which, erroneously in remote sensing, belong to one overly broad plant functional type, the sphagnum mosses. The position of each sphagnum moss species is relative to the depth to water, with sphagnum capillifolium occupying high hummock positions, sphagnum papillosum in intermediate positions, and our low rider sphagnum cuspidatum occupying the lowest positions along the hummock to hollow gradient. If cuspidatum is the hair in the foot base of sphagnum growth, then capillifolium would be the tortoise. This fast-growing hollow species gets an early lead in shoot growth, but in terms of actual productivity, it can't beat the slow, steady, and prolonged productivity of the capillifolium hummock species. These preferred eco-hydrological niches are the result of the previously mentioned adaptive functional traits that are species-specific. These traits include such things as water transport and water holding capacity, differences in decay resistance, also known as recalcitrance, differences in shoot density, and most importantly for us, differences in light harvesting mechanisms and specific adaptations to coping with high photic stress experienced in these open and unshaded ecosystems. Photic is referring to the natural light regime that these plants experience. This last suite of photic adaptations are what determine the species-specific spectral reflectance response of sphagnum. So basically, each species has its own reflectance response based on, its own physiology and anatomy. And that's what I'm actually here to talk about. So in terms of productivity in the ecosystem, everything is a function of PAR. PAR stands for photosynthetically active radiation, the part that plants actually harness for the process of photosynthesis. This is a diagram of the relevant sections of the electromagnetic spectrum. PAR is the region of wavelength of electromagnetic radiation, aka sunlight, that drive plant metabolism and primary productivity. This diagram here shows an example of a spectral reflectance profile for sphagnum papillosum. The visible and near infrared bands that you can see on the profile are shown, along with the vegetation fluorescence peaks, as well as the water reflectance trough typical of sphagnum mosses. So what we basically see here is a spectrotype. So, each species of plant will have its own, spectral profile specific to itself. In terms of gross primary productivity and carbon assimilation, peatlands are characterized by substantial amounts of organic matter, with stocks commonly ranging from between 50 to 150 kilograms of carbon per square meter, and then accumulation rates over a year ranging between 20 to 30 grams of carbon per square meter. These accumulations represent an imbalance between the input of carbon from plant production and carbon export from the peatland as either methane, carbon dioxide, or dissolved organic carbon, which are the products of the decomposition of plant tissues and organic matter in the soil. So this is just showing all the different pathways that it leads. So what we've seen is a simplified diagram of the carbon cycle in a peat bog. Looking at the left-hand side and the bottom of this diagram, you can see the catothelm and the acrothelm labeled. These are just layers. The catothelm is deep peat, which remains saturated, while the water table level varies within the acrothelm. Shifts in photosynthetically active radiation, PAR, are strong determinants of sphagnum productivity and the assimilative capacity of the peatland. The net ecosystem production of peatlands is the difference between this gross CO2 assimilation via photosynthesis and the total ecosystem respiration, which is the sum of both autotrophic and heterotrophic respiration. Both sufficient water availability and adequate light, PAR, determine the assimilative capacity of sphagnum. At PAR readings of roughly 200 micromoles per meter squared per second, net ecosystem production for peatland approaches zero. But with increasing PAR levels, these systems become net sinks for CO2. So even at quite low levels of light, these become sinks. So they're sequestering more carbon. Rates of net primary production of peatlands are generally small, as are rates of net ecosystem exchange of carbon dioxide and soil respiration. Due to this, the accumulation of organic matter in peatlands generally comes down to slow rates of decomposition, not rates of production. These slow rates of decomposition are associated with the cool temperatures, the anoxic conditions, so the lack of oxygen in these waterlogged areas, and then the functionally limited decomposer communities. So there's not as much nutrients in it, so you don't have the same sort of decomposers around. And then also the litter and organic matter substrates that are naturally slow to decompose, like the sphagnum. The litter of hummock-forming sphagnum species is dense with what are termed phenolics, which add to this decay resistance. So what doesn't break down accumulates, and that's how we get these very long-lived ecosystems storing so much carbon. A carbon flux study from an adjacent area of Burns Bog that looked at the gross ecosystem productivity was highest between April and June, followed by a late growing season from July to September. This flux tower eddy covariance study of ecosystem carbon uptake would not be expected to define the productivity cycles of sphagnum plants. These sorts of flux towers are very common in peatland studies, but as you'll see in the rest of the study, the models and stuff that we base on this and remote sensing aren't necessarily that reliable or accurate. Our study took on a recommendation to expand the time frame of sphagnum monitoring throughout all potential growing seasons, not just the vascular one, which we can refer to as the phases of productivity. So from a master list of bryophytes, mosses, put together by Dr. Karen Galinsky, a sphagnum expert, the 3 indicator species of sphagnum were chosen based on their representation of their functional positions along the hummock to hollow gradient. It's important to remember the key features of sphagnum that shape the habitat and drive peatland dynamics. They create an environment that is acidic, nutrient poor, wet, and anoxic. They tolerate and require low concentrations of nutrients. They are resistant to decay, and there are a number of species that specialize in different parts of peatland gradients. These specialists have unique functional traits that include differences in decay resistance and water transport and holding capacities, the two sides of the productivity coin in terms of carbon productivity. How non-vascular ecosystem engineers access and utilize moisture and to what degree they are decay resistant is central to peat forming processes both locally and at the landscape scale. Moisture-mediated sphagnum productivity is the linchpin of carbon assimilation, which is inherently linked to decay gradients, as the same species that have superior water transport and holding capacities also have higher rates of decay resistance. This is all a long-winded way of saying that along this hummock to hollow gradient, species have adapted traits that affect their net primary productivity. Although some faster growing species seem more productive, they have attenuated productivity windows, and they also are more susceptible to decay and decomposition, readily releasing the carbon they assimilated during the less ameliorative moisture conditions. This photo here is of an extensive sphagnum cuspidatum hollow that has entered its senescence phase during the summer drought period. The resumption of moisture-mediated photosynthesis will arrive with September and the first fall rains, when the local water table begins to rise from its summer maximum. Certain species have prolonged productivity windows, especially densely packed and high-growing hummock species. These species can have extended periods of productivity well outside of the vascular growing season. This hummock here is still productive in its upper canopy after the frost had set in late November. So it's still assimilating and sequestering carbon and being productive plant well after the frost has set in, so well outside of a vascular growing season. So knowing that photosynthesis equals primary productivity, and that for these non-vascular plants, photosynthesis is moisture mediated, we targeted primary productivity by measuring spectral reflectance along the hummock to hollow gradient. Sphagnum are the keystone species of the ecosystem and also of the plant assemblages along this gradient. Again, peat mosses are not all created the same. They have these physically different roles, and niche partitioning is based on the suite of specially adapted functional traits for each species. In the study, I was interested in the phenospectral niches of the 3 different species along the gradient at my study site. It was these phenospectral niches that we were looking to investigate and hopefully identify. When I use the term phenospectral, I'm referring to the reflectance of light from a plant, in this case sphagnum canopies, based on each plant's particular phenology. Phenology being the study of periodic events in the biological life cycle of an organism, and how these events are influenced by seasonal and interannual variations in climate. In the case of the specific phenologies we are interested in, were the plant-specific cycles of moisture-mediated senescence and rejuvenescence. If these were vascular plants, I could say green up and die back, but it's not recommended to map vascular concepts onto non-vascular plants. So what I was interested in was monitoring sphagnum productivity for the 3 indicator species occupying the distinct niches. Why? Because the central measurement of ecosystem integrity in a domed community, in the domed community of a raised bog, is peat accumulation as a result of this sphagnum productivity. So how did we determine the productivity of sphagnum? Quite simply, were the sphagnum canopies photosynthesizing? Yes or no? The hummock on the left, photosynthesizing. The hollow on the right, not so much. Again, photosynthesis in these non-vascular plants is moisture mediated. So it's the hydrologically induced trends in productivity that are most interesting. It's these trends in reflectance response of sphagnum canopies that we monitored in tandem with the locally immediate water table. And yes, for the study of bryophyte photosynthesis, so bryophytes being all mosses, the unit of study is the sphagnum canopy. Individual shoot productivity or photosynthesis is not studied, nor is leaf area index a useful metric. Because of their non-vascular quirks and the resulting morphologies of these plants, the scientific consensus has established the canopy as the accepted unit of study. I am not a bryologist and I don't make the rules. I just have to kind of foreground that, because often I'll run into colleagues and stuff who work in forest and they're like, it's not a canopy. It most certainly is a canopy. That's how it's treated. When I refer to the sphagnum canopy, think densely packed, moisture-retaining and acidic hummocks and moss carpets, like in these 2 images. So you may be questioning why we need multispectral imaging when we could just use traditional red, blue, green imagery to monitor sphagnum. Traditional images are much like our eyes, but your eyes don't perceive color, not exactly. Your eyes perceive light, specifically light with wavelengths between 380 and 740 nanometers, what we refer to as violet and red. Remember that photosynthetically active radiation, PAR, designates the spectral range for solar radiation from 400 to 700 nanometers, the waveband of light that photosynthetic organisms use for photosynthetic processes. This waveband roughly corresponds with the range of light visible to our human eyes. Light stimulates the cone cells in our retinas, and from there our brain takes over the perceiving, hence dress-related domestic disputes. Isaac Newton first showed in Optics of 1704 that color is not an inherent property of an object, excepting those that are luminescent. Color is instead the effect of an object's reflection of certain wavelengths of visible light and the absorption of the rest. That's the reason that although I can see the difference between these 2 interspersed species of sphagnum in the image, my visual system is not the most reliable tool for the assessment of changes in color and reflectance. My eyes' red, blue, and green cones actually provide me with my own personal false color rendering of the moss, but it's limited. As with a traditional red, blue, green photograph, a lot of valuable information is lost. So instead, we rely on multispectral imaging, which extends our visual sense while at the same time making it discrete and quantifiable. So what are we sensing? We're sensing the moisture-mediated productivity of peat mosses after hydrological restoration and the natural regeneration of the acrotelm and the topmost layer of peat, the pectotelm. So beginning in 2001, ditch blocking was initiated in Burns Bog to decrease water drainage and restore stability to the local water regime. In 2012, to peat and chloroplast. So chloroplast, picture like a lawn sign during an election. So dams of peat and dams of chloroplast were installed in 2012 in drainage ditches near our study site. There are now more than 225 handmade dams in the bog, all aimed at retaining rainwater in this ombrogenic environment. Ombrogenic just meaning its genesis is from rain. Ombro meaning like umbrella, rain. To give you a better idea of what's involved in this ongoing restoration work, I'm going to defer to Don DeMille, the architect and builder of the lion's share of these dams. So here's a short video just showing you what they do.
Don DeMille is a guy who really gets into his work.
Oh, that makes a world of difference when you go up.
Don is contracted by the Corporation of Delta to build dams in Burns Bog. It's his work site, but its waters also provide respite from this brutally physical work on a scorching hot day.
I actually started building dams in the bog while it was still privately owned. In 1996, there was a massive rectangular canal dug in the very center and the very heart of the bog, and it was really, really damaging it. So we built some quite big dams in there that, you know, really held back the water a lot. Let's use this monster here.
We're really lucky to have Don DeMille in our community. He's very, very passionate and dedicated to trying to preserve the bog.
Decades later, large pieces of industrial equipment sit rusting in the bog. From the air, you can clearly see where large ditches have been dug to facilitate the harvesting of peat, an industry that started here in the 1930s and continued till 1984.
This is unusual for snow in the bog.
Today, the water in Burns Bog is key to its restoration.
So the first one we're going to do is measure down to the water table. So we've got this on. So water is basically the lifeblood of a bog. A raised bog like this is actually managed to bring itself up above the groundwater table. And so it's only fed by precipitation, rain, fog, snow, things like that.
There are now 225 dams in the bog, all aimed at retaining rainwater. Beaver dams play a role too. They are dotted all over the wetter areas of the bog.
Sarah, I'm going to take you down to where I did a dam 3 or 4 years ago.
But human intervention here, closely supervised by the Corporation of Delta, is absolutely crucial to the restoration of the bog.
It was a place more of mystery and wonder, and now I've tramped the whole thing and I haul equipment in, so it's lost some of its mystique, but it's still wonderful. There's always something neat. I got about 25 or 30 feet from a pair of cranes last week, and always something cool, something different. Eagles flying over most of the time, hawks. Even just in here, a fly lands on your shoulder or your wrist, and you have a look at it, and it's usually something really weird that you've never seen before because there's such diversity. There are something like 300 to 500 kilometers of ditches that need to be dammed. I'll have to live to be about 500 years old to get it done myself. So it's just so gigantic.
All right, so that gives you a better idea of what rewetting and the actual like wholesale restoration of the hydrology of the whole bog kind of looks like and what it involves. It also gives some good flyovers of the situation of the bog. So thank you to Don and Dr. Howie for the informative video. So after rewetting of our portion of the bog between 2008 and 2014, water table levels monitored by the 3 piezometers, which are marked in white in this image, indicated a seasonal variability. So the water table obviously goes up and down with the rains and the precipitation that we experience throughout a year. And through this monitoring, it resembled the normal historical hydrological fluctuations that would have been experienced before all these incisions and linear features were made into the bog. So also, according to a site assessment of our cranberry fields carried out in 2014, hydrological conditions were no longer considered a barrier to restoration of peat forming processes, which are the most integral part of the restoration of the bog. So you may have noticed a recent act of bog rewetting that got international attention. On February 25th, the second day of the Russian invasion, Ukrainian forces released water from a hydroelectric dam on the Irpin River to block the Russian advance, flooding the village of Demyev. The village was flooded when troops opened a nearby dam, which sent water surging into the surrounding fields and bogs, which had been previously drained for dryland agriculture. This sudden rewetting created a literal quagmire, which thwarted the Russian tank assault on Kyiv, which provided time to prepare defense of the capital. I just wanted to mention that because I think it's one of the most interesting rewettings of a bog ecosystem that has happened. Tragic, but it did help protect Kyiv. So closer to home at our field site in Burns Bog, after ditch blocking and damming, the surrounding ditches have been colonized by the peat moss pioneer species, Sphagnum pacifica. So like other ecosystems, including upland ecosystems, even bogs have their own resident pioneer species, who are the first to arrive in succession. So the recovery of the water table to within historical bounds was an important threshold that had to be exceeded before natural regeneration could take place on this cutover surface. Spontaneous dispersal of sphagnum diaspores and peatland nurse plants seem to have activated an ecological priority effect on the once exposed catatomic peat, that catatomic peat being just the muck underneath in the anoxic zone. So nucleation and infilling along the formerly flat surfaces seem to be leading to the site on a trajectory towards a more typical hummock to hollow complex. So this is natural regeneration after getting rid of that coarse impact of degradation, being those incisions into the bog that kept water out of the peat mound. So knowing all this, we set out to monitor the reflectance response of the 3 indicator species of sphagnum using 3 spectral vegetation indices that are sensitive to moisture-mediated sphagnum photosynthesis. I know that's a lot, so I'll try to unpack that. Spectral vegetation indices, what are they? So these are the spectral vegetation indices, SVIs, that we chose to rely on. So it's the normalized difference vegetation index, probably the most common in the world. It's related to the abundance of photosynthetically active tissue and compares the maximum absorption feature in the red region with a reflection of near infrared reference value. So it's comparing 2 parts of the electromagnetic spectrum. Then we have the PRI, the photochemical reflectance index, and it's an indicator of photosynthetic light use efficiency and targets reflectance associated with the photoprotective states of certain photoprotective pigments in the moss. And then we have the chlorophyll index, which targets reflectance associated with the edge of a previously identified chlorophyll absorption feature that's centered on 705 nanometers. So, as one colleague rightly pointed out, these reflectance indices that I've chosen are associated with photosynthesis. And they wondered, why, if I was interested in local ecohydrology, meaning the way that water and the plants are interrelated, the ecohydrology, why, if I was interested in that, was I looking at these regions of the electromagnetic spectrum? Short answer, because sphagnum are ectohydric and poikilohydric. And being non-vascular plants, they require adequate moisture levels to carry out photosynthesis. Being ectohydric, sphagnum mosses cannot rely on internal means of water and nutrient transport. They don't have vascular tissue. Being poikilohydric, they're unable to actively regulate uptake and loss of plant water. So due to this, the spatial distribution of different species of sphagnum along gradients is ecophysiologically mediated. The microtopographical position along hummock to hollow gradients is a function of the local moisture regime. So the return of sphagnum productivity after water deficit periods is moisture dependent. So in vitro and ex situ studies, so at other sites and in the lab, research has shown a link between these 3 indices and moisture mediated photosynthesis. So that answers the question of this colleague. So I keep saying so a lot, sorry about that. These earlier studies were either carried out under controlled conditions, or they focused on fewer species and fewer indices, or they occurred over extremely limited periods of time. Due to these constraints, what relationships were found could be characterized as being sample specific. So they don't represent the population of moss, they were just representative of the sample that the researchers looked at. I wanted to avoid sample specificity in our research. I wanted to investigate and determine suitable temporal windows, so windows of time, that would be the most meaningful under variable hydrological conditions. And I also wanted to assess for the potential of these indicator species to be differentiated in this raised bog. So the concentration of photoprotective compounds in sphagnum has been used as an indicator of changes in photosynthetic activity. Narrowband multispectral imaging captures light from wavelengths associated with the underlying concentration of these compounds, and this serves as a proxy for the photosynthetic activity of the sphagnum. Narrowband indices can be used to monitor both ecophysiological functions of the plant and to capture species-specific spectral profiles used in plant identification. Earlier, we'd seen an image of a spectral profile for Sphagnum papillosum. This is like the spectral ID. So everything that you can shine light on or get a reflectance off of, whether it's the surface of Mars or a plant or barren rocky soil, will have its own spectral profile. Although species can be distinguished by human observers in the visible portion of the spectrum, tracking reflectance features in the near infrared and beyond requires technological assistance. In situ spectroreflectance studies that employ, so at the site that you're looking at, that employ multiple indices and multiple indicator species of sphagnum along the gradient are really scarce. Before spectrally assisted monitoring could be upscaled to the landscape level, certain questions concerning the specificity and reliability of these species-specific responses should be investigated. Although useful technologies exist, knowing their limitations and capabilities is important to understanding their potential applications for peatland monitoring. This will harken back to the first slide that I showed where we were looking at the MODIS satellite-based imagery of NDVI. So this study that we carried out was trying to look at the reliability and the veracity of what these satellite-based platforms are often saying. Establishing whether moisture-mediated spectral signatures exist for local indicator species of sphagnum was a primary objective of the study, as was the testing of indices responsive to sphagnum photosynthesis along these gradients. We hoped that lessons learned during this observational study could contribute to the development of future light touch peatland monitoring. So that could be, light touch monitoring methods that could include drones flying over that could identify species and could identify the depth to water table. As well, we thought this could maybe form the foundation of what are termed regionally specific phenospectral libraries. So understanding the spectral reflectance of different species over time throughout a year, hence phenospectral. What we're looking at here is a map showing the relative locations of our spectral imaging plots across the study site. The colors correspond to each species. Multispectral and hydrological data were collected from 25 species-specific canopies with individually installed dip wells or piezometers. During the installation of 48 additional permanent vegetation plots for another study that I carried out, the entire study area was traversed, and this gave us the opportunity to select imaging plots that were most conducive to long-term monitoring. These plots were chosen for their monospecificity, which helped limit spectral mixing, preserving spectrally pure reflectance signals. So monospecificity means that they were homogeneous. It was just the moss, so we could really hone in on the signal from just the moss and not overlying plants. In total, 40 field trips were made to the study site in Burns Bog, with 24 trips devoted solely to multispectral imaging. Unlike other multispectral and hyperspectral peatland studies, field days were not limited to the vascular growing season, which allowed us to capture imagery during favorable conditions for sphagnum productivity. This helped us to avoid the vascular bias associated with poorly resolved remote sensing studies, resolved referring to the resolution of both the resolution of the study in space, time, and spectral resolution. Again, avoiding phenospectral bias requires an understanding of the phenology of those species that are central to the ecosystem in question. And that would apply to any ecosystem where you're trying to use, spectral imaging. All multispectral imaging was timed to coincide with full solar illumination, with all measurements taken during a 4-hour window centered on solar noon. Field visits for image acquisition took place in every season under variable conditions, establishing an image diversity that incorporated and captured the natural morphological and physiological differences. To ensure a stable and uniform viewing geometry, a light and portable frame, which you can see in this image, was fabricated based on a design that I borrowed from a near-shore monitoring study. Two 4-meter lengths of rebar were installed at opposing diagonal corners for each imaging plot. These held the base of the frame stationary and ensured proper orientation was maintained throughout the entire study. Images of each moss canopy were taken from nadir, meaning from above, with coincident measurement of PAR to account for changes in solar conditions during field days and over changing seasons. All imagery was standardized to account for differences in irradiance using references images from a standardized reflectance panel. That's a bunch of, science speak for we wanted to do our homework and make sure that this was verifiable and there was a veracity to whatever we found. The Tetracam multi-camera array that we used to capture stacked images from 6 narrow bandwidths, with each of these images synced and georeferences. What you see here is the first channel in the larger image, and then you see the other channels down the right, or bandwidth from a stacked image. Along the left-hand side, you can see the other images derived from the same scene, each utilizing the reflectance values from one of the other 5 channels. Each pixel for each band contains values of between 0 and 255, depending on their brightness, i.e. the intensity of reflectance from that pixel at that wavelength of light. So each image down the left is a different wavelength, and then I can hover over them and make them the larger image. And what you're seeing is the actual reflectance value from each discrete wavelength. Having 6 channels allowed us to extract the ratios of light needed to derive the spectral vegetation indices, the SVIs, that we were interested in. Because we wanted close range spectral products, we selected a depth of field of 0.7 meters. This provided us with a ground field of view of a half square meter. Due to this, each pixel in these images corresponds to an area of moss of roughly a half square millimeter. The focal length of the camera had to be adjusted to accommodate near sensing at such a close distance. These images are in grayscale, and we only adjust them with false coloration to assist human identification and differentiation of features in the scene. The index values that we calculated for the normalized difference vegetation index, the photochemical reflectance index, and the chlorophyll index are derived from these grayscale raster image stacks. I know it sounds confusing, but all we've done is taken 6 pictures of each individual canopy, and these 6 images are each perfectly synced both in space and time. What you're seeing here are data sheets from throughout the study period. These spatially and temporally synced images allow us to use fairly simple maths, compare the ratios of light reflected at different wavelengths for the same tiny fraction of the canopy or for the entire canopy. These ratios of reflectance intensity gave us a spectral vegetation index values that we use to track changes in reflectance over time. We then use these spectral vegetation index values to compare the reflectance signal from the entire field of view with the reflectance signal for the species-specific region of interest. So we're comparing 2 spatial scales. We also use these reflectance values to statistically discriminate between species. Can we see differences between species? And this species discrimination was accomplished by means of the relationship of the underlying water table to the moss reflectance. The close monitoring distances selected for this observational study were based on the scale of diffusive and photosynthetic processes at the peat-air interface, where water, sunlight, and gas meet to sequester carbon. The isolation of a distinct region of interest within the larger field of view allowed for a crucial comparison of spectral response at 2 fine spatial resolutions, the decimeter and the sub-centimeter. While plots have been selected in other near sensing studies due to ease of access, amongst other logistical reasons, plots in our study were selected solely for the suitability to reproducible imaging of species-specific regions of interest. It was this comparison of 2 fine spatial resolutions that allowed us to assess that spatiospectral uncoupling. And it was the collection of imagery of the depth to water measurements throughout multiple periods of productivity that allowed us to assess phenospectral bias in the remote sensing products. We set out to monitor moisture-mediated reflectance across 2 vascular growing seasons, incorporating multiple cycles of sphagnum productivity and dormancy. The response variables included the 3 indices, and the explanatory variables included depth to water, indicator species, and the 2 close range spatial scales. We used multiple linear regression and linear effects modeling to capture the relationship between these variables. We ran correlation tests for each possible relationship. We established the statistical significance of these relationships, and we checked their strength. In recognition of the complexity of these systems, we adopted thresholds of statistical significance of 0.001 for very strong evidence and 0.005 for strong evidence. So we threw away the idea of 0.05 for, showing a p-value where we go, oh, it's statistically significant. No, this is a very complex ecosystem, so we wanted to step up the game in terms of significance. So what do we find? Taking the grayscale imagery that I've just showed you of the moss canopies and applying color palettes, we can track the changes in indexed response. Because NDVI performed best, the normalized difference vegetation index, I've chosen one typical plot from each species to share. Okay, we'll look at Sphagnum cuspidatum, the hollow species, using what's termed a discontinuous color palette. This type of palette helps in the discrimination of divergent spectral patterns, meaning they can help in the identification of vegetation types that have divergent spectral phenologies. So all of those grayscale values, we've just applied a color to. So see the discontinuous color-coded index values at the top right. That's giving you NDVI values. Higher values mean it's more productive, means it's photosynthesizing, means the plant is actually assimilating carbon. And then what you see in the bottom is depth to water table graph, and then the circle shows you which date each of these images come from. What we notice is that the NDVI values have gone up as depth to water is decreased. This hollow is still experiencing moisture conditions conducive to sphagnum photosynthesis. Come July, NDVI values have entered unproductive territory. The canopy, the sphagnum moss canopy, is senescent. It's not producing. But with September, the resumption of photosynthesis follows the first rains. Although the underlying sphagnum is productive, the overlying bog cranberry is not. So you can actually see the bog cranberry starting to grow out over the top here. Lower NDVI values attributable to vascular dieback will affect the average index value in a mixed spectral scene such as this. We'll now look at Sphagnum papillosum hummock, which has better water transport and water holding capacities than Sphagnum cuspidatum, the hollow species, despite the greater depth to water table distances. But in this time, we'll use a continuous color palette for easier productivity tracking. It helps if we keep an eye on the depth to water distance labeled on each image. So on the right, you can see that it's a continuous color palette, meaning that for our eyes, we see it as like the spectrum the normally way it would go. So in this case, it's from green to like a deep orange, almost red. Things are okay in June. Better in July, despite a drop in the depth to water, but there were those late rains, and this is expressed in a productivity lag. Depth to water distances have become too great and productivity has ceased. We can see this in the yellows. You can see that moisture mediated productivity lag in certain parts of the canopy after the early fall rains, but that changes as whole canopy rejuvenescence proceeds. So green up if this is a vascular plant. By November, the frosts have begun, but most of the canopy is still productive in this intermediate species. And now Sphagnum capitulifolium, this peat-forming powerhouse is the tortoise in that productivity race that I mentioned, slow and steady with longer phenophases of productivity and the ability to bank surplus carbon in decay-resistant plant residues. Still highly productive in early July. The depth to water for this hummock is 52 centimeters, yet parts of the sphagnum canopy are still photosynthesizing. Things shift. And then a productivity drop associated with a short dry spell. The lower lying areas have succumbed to frost as temperature threshold checks sphagnum photosynthesis. Now using a continuous color palette to track the changes of the same canopy. So just pay attention to the high greens to low yellows and how we go through the season and what month it is and where the water level's at. It's notable that even high and dry hummocks can experience oversaturation during the wet season. So of the three indices, NDVI had the highest correlation with changes in depth to water. The photochemical reflectance index performed poorly throughout the study, and the chlorophyll index experienced temporal lags in response to changes in depth to water. So we were trying to assess which is the best index to use. NDVI in the study seemed to be. In terms of correlation, our sample data did provide sufficient evidence of a relationship between depth to water and canopy reflectance in our population. The strength of these relationships varied by species, by the index that we used, and by the spatial scale. NDVI not only had the strongest correlations, but it was also the most responsive to changes in depth to water. We found that differences in the strength of the relationship between depth to water and reflectance response were based on species of sphagnum and the index employed. In terms of the statistical reliability of each index, NDVI showed very strong evidence for the relationship between the response and indicator variables across each species. So it's very good at discriminating. We are also interested in the effect that different conditions have on the relationship between changes in depth to water and reflectance response, specifically spatial scale and indicator species. So to assess the effect of moving between two close spatial scales, we used hypothesis tests to confirm that the differences between the regression coefficients and constants for both the field of view and the region of interest by species were statistically significant. In this case of the scale, moving between those two close spatial scales, we were able to reject the null hypothesis, the conclusion being that shifting between two close range spatial scales does affect the reliability of the relationship between depth to water and reflectance response. We also used hypothesis tests to confirm that the differences between the regression coefficients and constants for each species were statistically significant, making sure that the relationships we were seeing were species specific. Using the relationship between depth to water table and reflectance response, NDVI could reliably discriminate between each of the three indicator species of moss. So strictly speaking, we were not comparing differences in absolute reflectance between species. We were examining the relationship between reflectance response and changes in depth to water, with a comparison of this relationship carried out between each species. There is an important and subtle distinction between these two things. The indices we chose, specifically NDVI, don't just register differences between species-specific spectral response. They quantify these discrete differences. As a function of the natural range of depth to water variability, these species-specific differences in spectral signal were statistically significant. This is very important because it means we can actually track species and identify species in relation to one of the most important variables in this peatland, which is change in the local hydrological regime, the depth to water. We checked all of our model assumptions during regression analysis, and this speaks to the reliability of the relationships we found in our population. The evaluation of model attributes and results was guided by theory and subject area knowledge, avoiding automated procedures for model selection and specification. We left the math washing out. We focused on the theory behind the relationships we were looking at. We didn't use large data sets with machine learning algorithms. Our primary goal was to understand the nature of the relationship in our data. The establishment of a high R-squared value was not a direct concern of our study. Recognizing the complexity of this eco-hydrological system, we anticipated a high degree of data point scatter. Again, noise was expected, as it is acknowledged that depth to water table is not the sole potential predictor of sphagnum reflectance. It is believed that the precision of our measurements, including spatiospectral resolution, fit the target species and the scale of the underlying eco-hydrological processes as strong evidence for differences between both scale parameters and species held across both linear and mixed effects modeling. So the study would seem to suggest that with adequate spatiospectral resolution, near sensing can provide an information-rich representation of the hummock to hollow gradient, both during and outside of seasonal moisture deficit periods. Microsite sphagnum species experience different periods of productivity linked to their proximity to the water table. These distinct phenophases are linked to their respective positions along the gradient, and therefore their vertical distance from the water table. Field of view and scene resolution is a spatio-temporal question, and to avoid a dominant vascular spectral signal, the phenophases of dominant vascular plants along the gradient should be used to identify potential information windows that preserve the fidelity of sphagnum reflectance, if that's what you're monitoring. Periodic precipitation is necessary for sustained sphagnum productivity at low water table positions, meaning a little bit of rain in June like we're experiencing now. Multispectral imaging achieved at close range can differentiate between species of sphagnum and track the effect of the periodicity of precipitation on moss productivity. This is very important in a changing climate where the periodicity, i.e. when we are getting our rains and precipitation, is happening. We confirmed extended intervals of productivity for sphagnum in temperate raised bogs on this coast. We also confirmed winter growth and productivity. However, the most productive phenophases occur mostly in the spring and fall. The use of 2 fine-scale spatiospectral resolutions allowed us to examine the role that spectral noise plays at the decimeter scale for raised bog microsites. The link between moisture-mediated sphagnum productivity and peatland carbon balance is obscured when the spatiotemporal scales don't adequately capture and incorporate sphagnum phenophases. There's a growing need to develop monitoring methodologies that can efficiently and effectively track compositional changes along ecohydrological gradients within open site bog ecosystems. There's potential for the avoidance of either over or underestimation of microsites if multispectral imaging of indicator species were integrated into a classificatory scheme. Through the use of properly resolved and close-range multispectral imaging, the differentiation of sphagnum species is possible. The development of site-specific spectral libraries that incorporate multi-species and multi-temporal profiles is an important first step in this direction. In terms of capabilities of remote sensing and near sensing, it is important that monitoring methodologies that rely on near and remotely sensed technologies don't overpromise as they often have. Both of these images were taken in the spring of 2018. The image on the left was taken May 25th and shows one of the sphagnum capifolium imaging plots with bog laboratory tea plants clearly in flower. The image on the right, taken on June 3rd, shows a large sphagnum cuspidatum hollow with an overstory of bog laboratory tea plants also in flower. So if we have the spatiotemporal resolution of near and remote sensing in mind, so the size of those grids that the satellites can pick up, and when the satellites fly over and take images, what effect could the specific phenology of these bog laboratory tea plants have on the reliability of remotely sensed products from an open site bog ecosystem? This image was taken on July 21st, 2017. In this image, looking past the emergent Juncus species, maybe Juncus torii, we can see the onset of white-beaked sedge flowering. So again, if we have the spatial temporal resolution of near and remote sensing in mind, what effect could the specific phenology of white-beaked sedge have on the reliability of remotely sensed products from an open bog ecosystem? It's not just the divergent phenospectral reflectance profiles of vascular plants that can return errant spectral products at these intermediate and low resolutions. Although this high and dry sphagnum capifolium hummock is still actively photosynthesizing on October 3rd, 2019, the surrounding hollow is totally inundated. So inundated and saturated hollows can deliver errant reflectance values, and the subsequent interpretation of these reflectance values, especially if the resolution of remote sensing products results in pixel sizes that average out mixed spatiospectral scenes, like we saw in that very first slide. So this next photograph was taken at one of the field sites on October 8th, 2018. Notice the variety of coloring in the image. I will note that the spatiotemporal scale of the vast majority of peatland remote and near sensing studies that I canvassed during literature reviews didn't extend past the vascular growing season. From what I can recall, only 2 studies captured imagery, and this is from satellites, outside of the vascular growing season at their respective bogs. And this included just a single sampling day in September. Now, remembering the previous image of productive and photosynthesizing sphagnum capifolium canopy that was taken 5 days before this photograph, I think it's important to keep the specific phenologies of resident peatland plants in mind when interpreting both near and remotely sensed products. If monitoring the productivity of peatlands focuses narrowly on vascular growing seasons, and if the scale of the image capture leads to the previously mentioned spatiospectral uncoupling, then it's safe to assume that this would affect the reliability of peatland models that employ these near and remotely sensed products. As one of my literature review chapters on advances in peatland sensing outlined, there are issues with the accuracy and reliability of non-process-based peatland models that don't incorporate ecological expertise. Some have called for better process-based peatland models with greater characterization of species-specific vegetation phenology. One example of ecological expertise that would improve these models would be the incorporation of ecosystem-specific phenospectral knowledge. This incorporation would improve spatiospectral and phenospectral congruency. There are 3 scales at play in peatland sensing: the spatial, temporal, and spectral scales, and they all need to align. If they don't, then it is probably safe to assume that we wouldn't be asking the right questions. So thank you for your time and attention through all of that. And thank you to the 3 people on the slide. You can never complain when you're conducting your fieldwork with supportive and knowledgeable friends. and I definitely relied on these friends. And then also over the course of my research project, which took many years, I worked with multiple optical engineering firms, and nothing I did would have been possible without these partner scientists and engineers. I'd like to dedicate my research to Tetracam and the other scientific and engineering outfits that sadly did not actually make it through the pandemic. And that's it. Thank you.
Oh, that was absolutely incredible. Thoroughly enjoyed that. Wow. Absolutely wow. Well, thank you
Yeah.
so much for taking the time out of your day to come and give this little webinar. That was absolutely mind-blowing. Absolutely mind-blowing. Yeah. Yeah.
Too much stuff.
Oh, I love it. I love it. No, that's totally up my alley. I'm remote sensing major, so this is like,
Oh, okay. Sorry if I
Yeah.
slag remote sensing too much.
oh yeah, no. Oh my God. Stuff's complicated. So no, you nailed it perfectly. That was amazing. Oh my gosh. Oh my gosh. I'm like lost for words here. But yeah, that was fantastic. Yeah. Thank you so
Thank
much.
you.
And I know Izzy, who is the one you were liaisoning with, she's actually currently on the other side of the world, but I know she would be absolutely thrilled to watch this. I definitely think she's gonna tune in and watch as soon as I get this uploaded. But yeah, thank you again. Just incredible.
Cool. All right. Thank you. I appreciate
you.
that.